Hey, I just wanted to quickly plug that if you guys want any tutoring, some new slots have opened. If you look in the description, there's a link to my physics and maths tutor profile in which I do tutoring on there. Hello everybody, welcome back to another episode of the A-Level Cookbook. Today we're going to be doing the AQA A-Level Biology, paper 1 from June 2017. So it says, give two types of molecules from which a ribosome is made. Ribosomes are made of RNA, so ribosomal RNA, first of all. And also, it's made of protein. So, proteins. Next it says, describe the role of a ribosome in the production of a polypeptide. Do not include transcription. The entire point of transcription is to make mRNA. Translation is to then go and use that mRNA to make a protein. So what happens is your mRNA binds to a ribosome, like so. So here's your ribosome, here's your mRNA, blah, 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 blah. Here's it being read. What happens is you want to join amino acids together. Remember, that's the whole point of making proteins. That's the whole point of translation. So what you do is you've got this tRNA, which I'm just going to draw as this blob. I know it's clover-shaped and all that, but whatever, right? And what it'll do is it'll have an amino acid attached to it. The way it knows which amino acid to put where is because this tRNA will use its anticodon and form a complementary base holding it like this. Then the next tRNA comes along next to it for the next three, and it sits with the amino acid like this. They get joined together like that with a condensation reaction forming a peptide bond. And then the first one detaches, this next one stays. And then the next one joins up next, sits next to the amino acid, and then the condensation reactions happen like that. So it's literally just like that, step by step, until the entire thing is done. mRNA binds to the ribosomes. What happens is that tRNA with complementary anticodons forms complementary base pairs with the mRNA codon. And this brings an amino acid with it. I'm going to write A for short. Then what happens is, another one does the same. So I'm just going to write another one does the same. I'm lazy, but you write the whole thing. And then what happens is a peptide bond forms between the amino acids via condensation reaction. Every time you're talking about bond making or breaking, include what bond is broken and what's been, by how, condensation or hydrolysis. This is via a condensation reaction. And then it will move along to the next codon and so forth. So then it moves along to the next codon and the cycle repeats. There you go. Step by step and you'll be fine. So table 1 shows the base sequence of a part of a pre-mRNA molecule from a eukaryotic cell. Complete the table with the base sequences of the DNA strand from which this pre-mRNA was transcribed. So it's going to be whatever's complementary, but on DNA, right? So A goes at T, C goes at G, G goes at C. A goes at T. U is what replaces T, remember. Uracil replaces T in mRNAs. So that means this would have been T, so that's A, that's A, that's T, that's A. Next it says in a eukaryotic cell, the base sequence of this mRNA might be different from the sequence of the pre-mRNA. Explain why. It's saying what is different between this versus this. The only difference is that the introns have been removed by splicing to make mRNA. So then in mammals, in the early stages of pregnancy, a developing embryo exchanges substances of the mother via the cell of the uterus lining, the lining of the uterus. At this stage, there's a high concentration of glycogen in the cells lining the uterus. Describe the structure of glycogen. So it is alpha glucose, and it is a polysaccharide, joined by glycosidic bonds and branched. There you go, that secures every mark possible. During early pregnancy, the glycogen in the cells lining the uterus is an important source of energy for the embryo. Suggest how it acts as a source of energy, and it says do not include detail about the transport here. So this one, people started talking about lots of things to do with like, oh, there's branching and it's got lots of ends and this and that, blah, blah, blah. And it says here, it's indicative of students rushing to write everything they know rather than actually just listening to what the question says. All the question is saying is, how is glycogen a source of energy? So you would say it is hydrolyzed to glucose, which is used in respiration. That's it. That's all they care about. They know that you're going to try and rush to writing everything. It's just, there's no point writing all that. Suggestion explain 2 ways the cell surface membranes in the cells lining the uterus may be adapted to allow rapid transport of nutrients. So whenever you're transporting anything, whether you're diffusing and all of that stuff, right? Rules are to make it as fast as possible. You want a steep concentration gradient. You want a short diffusion pathway. You want a decent temperature, but obviously we can't really make our temperature go really high, so you can't always do that. A high surface area to volume ratio. And also, if you need facilitated diffusion, you need lots of proteins for it, like channel and carrier proteins. Whenever you have any question to do with the movement of substances, like gases and all of that kind of stuff, gas exchange, fish kills, all of that, rather than sitting and memorizing like 2,000 different things, you can just look at this and be like, oh, well, what do I know about, for example, fish gills? Oh, I know they have lots of capillaries and they're all one cell thick, and the mammalian gills and stuff are one cell thick. So there's a lot of surface area to volume ratio and short diffusion pathway and so forth. It's like, use these as the cues to then decide what you're going to say rather than just memorizing facts. So here then, we're looking at the movement of glucose and stuff, which requires channel proteins or carrier proteins, right? So in that case, one of the adaptations they must have is that they've got a large number of channel or carrier proteins, either's fine. And you need to say why, because a lot of people just say the feature and then don't really say like why. It's 4, so that increases, sorry, facilitated diffusion, right factor for short. In the question, it's only asking about the cell surface membrane, so we need to focus on the membrane stuff as well. So channel proteins are found in the membranes. We need to think of anything else to do with the membranes. Diffusion distances of membranes are always thin, so you can't really use that point. Here, it must be something like maximizing its surface area, so you could just, say that. The membrane has a high surface area to volume ratio. Why? It's so that you have more facilitated diffusion as well. So it's asking specifically about the membranes. It's not asking about, for example, like, oh, it's one cell thick and things like that. Or a good blood supply, because it's not relevant. It's only talking about the membranes. So answer the question carefully. So in humans, the gametes join of fertilization. Every cell of this undergoes mitotic division, blah, 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 and it gives you that. The first division takes 24 hours, and then every division after that is for 8 hours. After 3 days, it has a total volume of this. What is the mean volume of every cell? So we've got the volume. We need to find out the number of cells. So, okay, so for this question, we got the total volume of this entire thing. We need to find out the volume of every cell. So what we need to do is we need to figure out how many cells they got. So right now, at the very start, they have 1 cell. And then after this one division is 24 hours, and now they have 2 cells. And then from those 2 cells, you'll have, subsequent divisions every 8 hours. So then, they've told you it's 3 days, which is 72 hours. One division took 24 hours, so 72 minus 24 is 48 hours. That 48 is left for the other 2, the subsequent divisions. So 48 divided by 8 means you've got 6 divisions now. So therefore, in total, you've got a total of 7 divisions happening from the first cell. So it's going to be 1 times 2. That's your first division, times 2 times 2 times 2. So it's going to be 1 times 2 to the 7, which gives you 128 cells. So the volume is going to be 4.2 times 10 to the minus 3 divided by 128. 4.2 times 10 to the minus 3 divided by 128 gives you 3.28 blah 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 times 10 to the minus 5. We try and match the number of significant figures in the question. So we try and match the number of significant figures in the question. So it's going to be 3 point to 1 decimal place of 2 significant figures. So it's 3.3 times 10 to the minus 5 there. So here it says sodium ions from salt, sodium chloride are absorbed by the cells lining the gut. Some of these cells are membranes of the carrier protein called NHE3. NHE3 actively transports 1 sodium into the cell in exchange for 1 proton out. Right? So here's your cell. Na plus is in, but every time there's an H plus out. It says use your knowledge of transport across cell membranes to suggest how Na3 does this. So transport across cell membranes, you should be thinking facilitated diffusion, osmosis, simple diffusion, active transport, right? Within it, we've got lots of different types. So the facilitated diffusion / you've got an active transport is probably leaning towards the active transport because they've told you in the question, right? So active transport involves the use of co-transporter proteins, which are a type of carrier protein. So in this case, what's happening is we're using a co-transporter and ATP because, it requires energy. And how co-transporters work is like you've got your co-transporter protein here. It's quite similar to a carrier protein. So a carrier protein, something binds to it, changes shape, it lets it through, right? And then the thing can move right through like this. Okay? Co-transporter proteins, nearly the same. Only difference is 2 things need to bind to it, and then both of them can move through. But if we're active transporting it, then in order for it to change shape, then it needs an ATP. That's it. So here, what happens is the Na plus, and then the proton would be coming from the inside here instead. ATP is hydrolyzed, it changes shape. Sodium moves in, H plus moves out. So Na plus and H plus, or the proton, bind to, I'm going to write CT for short, the co-transporter protein. You write the whole thing. And then what happens is it changes shape. And then it just allows the movement. There you go, done. So scientists investigated the use of a drug called tenapanor to reduce salt absorption in the gut. This inhibits NHE3, which remember is Na plus in, H plus out. So generally speaking, whenever you're doing these experimental questions, think about what their dependent and independent variables are, right? And what they're actually measuring. So they're doing a high concentration of salt to both groups, but one has no, so the independent is, you know, tenapanor or none. And then the dependent is what they're going to be measuring. Let's have a look. So A was given no tenapanor, B was given some tenapanor. Both of them, they had their gut contents of the rats removed and then weighed. So then it's the mass of the gut. And the reason, what does this represent? Well, we need to figure that part out. When you don't give them tenapanor, it's this. So it's 0, but when you get, and it's 2. But when you give them tenapanor, it's gone up to 4. So tenapanor seems to have increased the gut of the mass of the gut of the content. Then it's told you as well, they did a statistical test to see if the difference in the means was significant, and they got a p-value of less than 0.05. This p-value is testing the difference in the means. That's what all p-values test, is something, and you need to say what, you need to identify what it's testing. Not just say the results are significant, you get no marks. You need to say that here, this is testing to see if there's a difference in the means that is significant. They concluded that tenapanor did reduce the absorption in the gut. Use all the information provided and your knowledge of water potential to explain how they got this conclusion. So start by describing what you see, and then you can put the gaps in. So they have said that you can see that tenapanor decreases the gut mean mass. So there's one start. Now you need to look back at this whole thing. Well, while we're here as well, this decrease is significant because the p-value is less than 0.05. Therefore, the difference in the mean content was not due to chance. It's what the p-value is testing. So like the difference in thing the p-value is testing is not due to chance. In this case, the difference in the means due to chance. All right. So then, what that means is because the probability of it being due to chance is less than like, you know, 0.05, right? So then that means that this gut has a higher mass. We need to figure out why. If you go back to the tenapanor in the question, it's told you that it inhibits the carrier protein, which moves it into gut cells. So if it inhibits that, then that means there must be Na plus in the gut, not the gut cells, in the lumen of it. So that means there's more Na plus in the gut. And then the question's told you to link it towards, you know, water. So you should be thinking, water potential. There's more Na plus in the gut. That decreases the water potential. Therefore, water moves into the gut down a water potential gradient via osmosis, and that increases the mass. There you go. So it's just looking at the question and what it's saying, because a lot of people were just sort of yapping about all kinds of stuff, and a lot of people didn't even successfully like interpret the p-value. You can't just say results. You need to describe what the p-value says. This p-value is less than 0.05. It is testing the difference in the means. Right? Always identify what the p-value is testing and scribble it down if you have to. High absorption of salt from the diet can lead to a higher than normal concentration of salt in the blood plasma. This can lead to a buildup of tissue fluid. Explain how. So he's quite literally told you that more salt in blood equals more tissue fluid. So you need to look at how tissue fluid normally works and then be like, what's going on here? So is your arterial end, there's your capillaries, and there's your venous end. What happens here is the pressure is really high, so it squeezes out water and solutes like this. Sodium chloride is a small solute, so it can actually squeeze out as well. Then this forms tissue fluid. At the venial end, remember that you've just shoved all the water out of here, so the water potential inside the capillaries is lower, so some starts to move back in via osmosis in the capillaries. All the exchange is happening in the capillaries, not the arterioles and venules. So then, if we have more salt in our blood, we're shoving more salt out into the tissue fluid, which means the tissue fluid now has more salt, and the water potential of the tissue fluid is lower. So that means that less water is going to go back in here because the water potential gradient is less steep. So then you got more fluid, more water accumulating, so you got more tissue fluid, and that's exactly it. So then what happens, let's write that down, is that you have an increase in salt concentration in tissue fluid, which means you have a lower water potential. You cannot just say sodium, you have to say sodium ions, you know, if you're going to say sodium ions, but salt's fine. And then that means less water returns to capillaries via osmosis at the venial end. You can't say venules, it has to be the capillaries at the venial end. That means that you've got you've got more tissue fluid accumulating. Alternatively, you could say that another thing you could say is that you've got high salt in the blood, meaning water moves into the blood, meaning the pressure's higher. So then at the arterial end, you've got a higher pressure shoving more water out, so there's a higher tissue fluid there as well. So describe how bacteria divide. This is by binary fission. what happens is you replicate the circular DNA, and then the cytoplasm divides, and then you get 2 daughter cells with a single copy of the DNA loop, or circular DNA. Washing powders often contain enzymes for bacteria. These enzymes include proteases that hydrolyze proteins in clothing stains. Figure 1 shows the effect of temperature on a protease that could be used in washing powder. So again, remember, dependent, independent. So don't just look at this and be like, oh, it's this. Look at the actual data here. So the independent variable that they are changing is temperature. And they are measuring the percentage of maximum protease activity. Really important that you do that, because questions asking, evaluations of what you should control or this or that, blah, 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 will link back to these. So then also, when you do these, look at each line one by one for a second and just describe it to yourself. So I'm going to start with this 30-degree line. It is just still. It is just the same. It's the percentage of the maximum protease activity. If it's 100%, this is working at max. If you look at the 50, it starts off at the 100 and it's working maximum, but then it decreases as time goes on, the percentage activity with 50, but then it keeps decreasing and then it looks like it's starting to level off. So it might actually level off at about 30% of the original activity. And if you look at 60, it's going down, and then all the way down to 0. So this clearly means that it's not able to do its activity anymore. So protease has stopped working here, right? Cool. So then it says explain the shape of the curves of this. Well, describe the curve, then explain it. So do it curve by curve, okay? So in this case, if we look at the curves, so if you look at 50 degrees, it's decreasing activity over time. So I know you don't get marks for describing it, but it helps you formulate what your answer is, because you're saying the thing and then you have to explain what it means. It forces you to do it. So then we need to explain why. It must be that, why is an enzyme not working at higher temperature? It must be that there's denaturing happening. You need to explain that as well. So hydrogen, ionic bonds, and all that kind of stuff break. Therefore, the active site changes shape. It's no longer complementary to the substrate, which is proteins. So you get fewer enzyme substrate complexes. That would explain the 50, right? However, 60 degrees C is showing the same, but it's a sharper decrease. So this must be happening more. So it must be that denaturation is happening faster than at 50 degrees, right? Because you can see it's shot down to 0, whereas here, but we need to describe what's the difference there. Why? Why? Is because it's hotter. The only explanation for that is that you've got more kinetic energy, so there is more disruption of bonds, of the the bonds mentioned above, which you can write again, due to more vibration. Do it like this and you'll never miss anything out. Some proteases are secreted as an extracellular enzyme by bacteria. Suggest one advantage to a bacterium secreting an extracellular protease in its natural environment. Explain your answer. So the only thing that proteases do in this question is what they've told you, is that it digests protein. So then you need to think, let's have a look then. What might that be useful for? They digest the protein and then they absorb the amino acids, and then you need to explain why that's helpful though. It's for protein synthesis, for example, or making new cell organelles and things like that, or naming it. That's what it could be. Mammals have some cells that produce extracellular proteases, but they also have membrane-bound dipeptidases, and it says describe the action of membrane-bound dipeptidases and explain their importance. So a lot of people just don't really say they'll either describe one thing and that's it and leave it there. Like, describe the action. Number 1 is what they do is they hydrolyze peptide bonds. Remember, whenever you talk about breaking or making bonds, you need to say what it is, condensation, hydrolysis, and what type of bonds. So it hydrolyzes peptide bonds to release amino acids, right? But that doesn't explain why it's important. The reason is because dipeptides cannot cross a cell membrane, and then you wouldn't be able to, absorb amino acids and actually use them again. We always eat food, break it down into its components, and then take them back and absorb it and then reuse them. So you can't do that if it's a dipeptide. So that's why we have dipeptidases, to break them all the way down into amino acids and be able to absorb them and use them. So just remember to talk about both points. Scientists investigate the treatment of human bladder infections caused by a species of bacterium. The species of bacterium is often resistant to antibiotics currently used for treatment. They investigated the use of these new antibiotics to treat the bladder infection. The new antibiotic inhibits bacterial ATP synthase enzyme. Place a tick in the appropriate box which shows the reaction catalyzed by ATP synthase. So you're synthesizing ATP from ADP and PI, which means you're going to do a condensation reaction, which means you're releasing water, so it's got to be this one. The new antibiotic is safe to use in humans because it does not inhibit ATP synthase found in human cells, suggest why ATP synthase is not inhibited and why bacterial ones must be instead. So it must be that, well, they've told you that this is some sort of inhibitor of an enzyme. So it's probably going to be going for the active site or changing the active site of it. So if it's not affecting human ones, but only bacterial ones, it must mean that the active site of the human one is different. So then you would say that human ATP synthase has a different active site shape. Or you could say the antibiotic is not complementary to human synthase, but it's showing you that it's inhibiting this, and you know, the only explanation is that they're both, you know, an enzyme. One is human, one isn't. So the only difference must be that. The scientists tested these new antibiotics in these 3 mice groups, cRNA, so C is control, untreated, R is with the current antibiotic, C is and A is with the new one. So then they removed samples of the bladder of this bacteria, the bladder after these mice after treatment, and then estimated the number of bacteria. So dependent and independent. The independent is going to be your antibiotic, whether you've been given it or not, or, you know, whatever. Independent is what they are counting. So it is going to be the number of bacteria in the bladder. Always take a second to look at the table or data or figure or whatever and make sense of it. So control is nothing. Remember, they're untreated. This is current antibiotics. This is the new antibiotics. And this is the number of bacteria in the bladder. So with nothing, you have the highest number of bacteria. Then when you give them the current ones, it drops down. And with the newest one, it's dropped down as well. Now if you compare them between each other, this has the greatest increase in bacteria. This one has a decrease, but it's not as fat as this. Can you see it's from here to here? Whereas here it's from here to here, so the drop is fatter here. So the question is saying 25 milligrams for every kilogram. We need to convert the mouse mass, oh my god, that's a weird tongue twister, into kilograms, which is 0.03 kilograms because you're dividing it by 1,000. 25 times 0.03 is going to be 0.75, and there you go. Then it says calculate the percentage difference in the actual numbers of bacteria in group A compared with R, and the actual number can be calculated by the log 10 value by doing 10 to the x. So show your working. So it's the difference in the actual numbers of A compared to R. So we need to look at the actual log 10 numbers of A and B. So if you look at A, that's what, 1.1, 1.2, about 1.29, maybe. So if you look at R, 2.85. So then what you would do is to get the actual numbers, it would be 10 to the 1.29 and then 10 to the 2.85. Then the difference of these, percentage difference, would be 10 to the 2.85 minus 10 to the 1.29 over 10 to the 2.85. And you times it by 100, and that should give you your answer, which we will see. 100 times 97.2%. So the scientists suggest that people newly diagnosed with bladder infection should be treated with both the current and the new antibiotic. Explain why they did this. Use information to your knowledge of evolution of antibiotic resistance in your answer. So if you look at figure 2, you can see that this decreases it, and so does this one, but it's not 0. So clearly there's some bacteria surviving still here, which could be that they're just resistant to A, but we don't know if it's resistant to the old antibiotics here. So these are the standard ones, remember, that's the new ones. So it might be that these ones are left over, might actually be susceptible to the ones here. So then what you could do is you could use both and probably wipe out way more. So the first thing we can say is immediately is that new antibiotic, I'm going to write ABX for short, you write the whole thing, does not kill all bacteria. So that must mean that there's some resistance going on. So using both, so there must be, therefore, some bacteria are resistant. We need to talk about the consequence of that. They're more likely to survive and reproduce. And that means you've got increasing resistant bacteria. But, you know, this may not be resistant to our current antibiotic. So combining both kills more and reduces the risk of this happening. And there you go. Now, only 6% of people taking this exam only got 3 marks here because they just talked about all kinds of stuff. It was either that they didn't realize that neither of the antibiotics killed all of them, or they just didn't explain the consequences of it, and a lot of them just wrote descriptive things about their knowledge of antibiotic resistance and didn't actually delve into addressing the question. You just cannot yap randomly. You have to do what the question's telling you. It's not about how much can you just unload on a page. Model answers and all that garbage do not work. So 2,4-D is a selective herbicide that kills some species of plants but not others. It disrupts cell surface membranes, but the extent of its disruption depends on different species. Scientists investigate the effect of a 2,4-D on wheat plants and on wild plants a weed. Remember, independence what you're changing. So what did they do? They grouped plants into both species in glass houses. They put each of them in one of two groups, which they treated as followed. So the independent, is 2,4-D versus none. And then after them, they cut 40 discs in each group, put them in flasks containing the ionized water, and they calculate the disruption by measuring the concentration of ions released into the water from the leaf discs. So what they're measuring, the dependent, obviously it's tempting to be like, the effect of this, blah, 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 don't do that. The actual thing they measured was the concentration of ions. So then you need to interpret what that means. So more concentration ions means more disruption. So then the results are shown in table 3, and then they said the lowest significant difference, the smallest difference between the two, that would give you this. So make sure you make sense of what that means in your own words. Do not just yap. The lowest significance difference is the smallest difference between the two means. So it is the difference between means that would be significant at this. So that means that if the difference is smaller than this thing, it would give significant differences in means. So what I mean by that is if my difference between the two of these is 7 or less, then I have a significant result. So it's really important to make sure you understand what that means. So then have a quick skim through the table. So group W had water, H had 2,4-D, and the concentration of ions, what they did is they did it between wheat and wild oats. So between wheat and wild oats, 2,4-D had not very much of a big difference or an effect. Between water and 2,4-D, though, you can see there's way more ions in the water in wild oats than there is in W. The difference between these, so it's not significant, but between these two, that is way more, that's more than 10, so this is a significant difference. So then it says, give 3 environmental variables that should be controlled when growing the plants when treating them with these sprays. So a lot of people were just saying a load of random things. You can't really say amount or level or whatever. Here, the reason I said you should write the dependent and independent variable is because when you're looking at things like, for example, what should be controlled, what precautions need taking, what, evaluate questions, it should all tie into what screws this up or what screws this up. Remember that the concentration of ions is determining the effect of this stuff on the weed plant, and that's representing the disruption of things. So anything that screws up the concentration of ions or screws up, giving them 2,4-D and D whatever, right, is what's going to screw the experiment up. So everything else needs to be controlled. Absolutely, possibly everything. So then, if that's the case, and we're looking at concentrations of ions too, temperature's easily one you could get there. Soil pH is another one you could get. Other things you can talk about. So you need to think about what can mess up, concentrations of ions in the sample. So it's also going to be the concentration of the ions in the sample to begin with. Because they started it in soil, and then they took it, and then dissolved it, and then looked at how much, was in there. So if they all had differing amounts of ions in there, some more than some less, then you don't really have a very nice, stable baseline. So here's your baseline. More disruption means more. You want to keep that the same. Other things you can talk about is CO2 concentration, humidity, and the distance between seeds and plants, but I think these are probably the strongest. So then it says, evaluate the use of 2,4-D as a herbicide that contains wild oats as a weed. Use all the information provided. So it's saying, is herbicide good to get rid of wild oats? So you should reflect your answer around that. So you should always say, when evaluate, things that are supportive statements and things that go against our limitations. So supportive statements, well, if we look at wheat, right, and it has oats as a weed, so these are the wheat crops, and you can see there's your wheat crops, and there's your wild oats. Well, first of all, if you put the 2,4-D on wheat, there doesn't seem to be much of a change, so it doesn't really seem to be destroying or killing or disrupting it, so that's a good thing, because we don't want it to do that to the wheat. But with the wild oats, it is, and there's a significant difference in these two as well. So the first thing you can say is, well, 2,4-D increases ion concentration in the wild oats, but not the wheat. This increase is significant because the difference is, what's that, 45, 50, 25? 25, which is greater than 7. Because remember that, sorry, not 7, 10. Because that's the minimum difference you need in order for your difference to be significant, is what the questions told you. There you go. So then, this still doesn't explain why that supports it. You're just repeating back the data. This means that it suggests more cell disruption membrane, disruption, which suggests increasing, likelihood of dying, increased likelihood of death. Right? Cool. But then we need to look at some limitation. They're saying, is this a good herbicide to kill wild oats? I don't see anything to do with actual dead plants. So first of all, we don't have any evidence of the oat cells, the wild oats dying, Literally nothing. As well as that, again, like I said, the controlled variable is concentration of ions, and they're talking about the disruption of cell membranes and also the fact that it's killing them. Again, no evidence of that. All they've done is they've just said, look, the ions are higher. That might suggest it. Another limitation as well is that it's talking about, you can't really say like other species or yap about other stuff, because here, like, the context is telling you that it's wild oats as a weed. So you can't really go ahead and say, oh, different, blah, blah, blah. It's like, no. But the other thing, though, is that weeds will depend on other things around the environment as well. For example, competition with other things as well. So we have absolutely no idea or no evidence of, like, the environmental impact it could have as well. What if this thing does, kill off weeds and wild oats and stuff, and absolutely butchers something else that the oats can, competes with, and now the oats like, yo, I'm allowed to chill and I'm happy. So scientists incubated the flask containing them at 26 degrees and then gently shook them, suggest why they ensured the temperature remained constant and then why they were shaken. So with temperature, they're keeping it constant because, well, they're looking at how much the cell membrane is getting battered by the 2,4-D, which means stuff's going to leak out. Now, if they have a really high temperature or cold temperature, it's going to get more or less fluid, and it's going to affect its permeability. So then the temperature is controlled, so you have no change in the fluidity of the phospholipid bilayer. And then why have they shaken it? The only reason is because if they did, think about what happens if they didn't shake it. Well, they just sit at the bottom. So if they shake it, it's to ensure all leaf discs are exposed to water. Also, another thing I guess you could say is that the ions are diffusing out of them. So if you shake them, you're evenly distributing them as well. So you're keeping that concentration gradient nice and balanced out. Otherwise, it'll accumulate here. But I think that's a bit of a weird one to say here. Describe how phagocytosis of a virus cell leads to presentation of its antigens. Do those. The examiner report says people are yapping about all kinds of stuff which had zero relevance to the thing. Phagocytosis, it's been engulfed, it's been digested, right? So then what? So the virus is engulfed and digested because it's engulfed into a phagocytic vacuole and digested because lysosomes fuse and release lysozymes into it. You cannot say that the virus enters or fuses with the lysosome because that's infecting the cell. So here's your phagocyte, it literally surrounds it like this. Here's your phagocyte, it literally surrounds it like this, whoops. And it's kept it here, the virus, and then what happens is if a lysosome comes along here, fuses with it and releases it into there. If it took it into this cell or whatever, it's been infected. So then, what happens is the virus is hydrolyzed by those lysozymes. So then it's telling you the antigen's presentation. The only thing that stimulates an immune response is an antigen. Anti, attack against, gen, potential to generate. Like pathogen, pathos, disease, gen, potential to generate. So here, what's happening is that the viral antigens are presented on the phagocyte cell surface membrane. That's it, you don't have to talk about anything else. So then it says, describe how presentation of a virus antigen leads to the secretion of an antibody against it. So lots of people just started talking about how it'd be presented, this and that, and then just, fumbled and then said a load of random rubbish. So presenting it, it's been presented. Who's it been presented to? So what happens is you've got a helper T cell binds to the presented antigen via a complementary receptor, and then it gets stimulated and releases chemical signals. This question's asking secretion of an antibody. Yes, it activates cytotoxic T cells, all of this stuff, blah, 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 blah. This question is asking about secretion of antibodies, so we're focusing on the antibody bit. So then what happens is a specific B cell type, because you don't want to activate every single B cell in the universe, you want the right ones, type divides by mitosis to form clones. This is known as clonal selection. And this produces plasma cells, which are those identical clones. And what they do is they all secrete the same antibody. I'm going to write AB for short. And that's your monoclonal antibody. That's where the name comes from. Monoclonal, one clone antibodies. There you go. That's it. That's all they wanted. So collagen is a protein produced by cells in joints such as the knee. Rheumatoid arthritis is an autoimmune disease. In an autoimmune disease, a person's immune system attacks their own cells. Rheumatoid arthritis causes pain, swelling, and stiffness in the joints. Scientists have found a virus that produces a protein very similar to human collagen, suggests how the immune response to this viral protein can result in the development of rheumatoid arthritis. So lots of people were talking about just repeating things from the question, or just saying this, and this, and blah, 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 and just saying all kinds of rubbish. It's saying here suggests how the immune response to the virus can result in rheumatoid arthritis. And they have told you that this virus makes a protein very similar to the collagen. So then, what that means is the immune response in humans will make an antibody against this protein, but because it is similar to human collagen, that antibody is also going to bind to the collagen and destroy it. You can't really say attack. So you will say the antibody made against this viral protein is also complementary to collagen. Therefore, it binds, which leads to destruction of collagen. You can't just say, oh, it attacks, so this and that. That's what the questions told you. You need to say how it's attacking and by, why the similar part tells you that. So figure 3 shows 2 different ways of classifying the same 3 species of snake. So they can interbreed to produce fertile offspring. So X is based on the frequency of observable characteristics. Y is based on their genetic characteristics. So looking at it and be like, oh, it's this color and that. This is based on their genetics. So X versus Y, we can see here, here, and here. Right? These 2 came from this, which both came from this, and this split into these, the ancestor that becomes these 2, and this split into something that becomes this. People overcomplicate this more than they should. Here's your grandparent, like 1, so here's your granddad, here's your grandma. They both have your dad, and then he has a brother, so he's your uncle, and he has another brother who's your aunt, and then here is you, right? Let me just check I haven't accidentally made some weird family tree. No, that's fine. One of your ancestors is your dad. One of your other ancestors could be your granddad and grandma. The common ancestor of all 3 of these is either your granddad or your grandma. You share a common ancestor with all 3 of these of your granddad and grandma. So if we look here, we can see that this is a species, this is a species. They both branched off from this point. So we have a common ancestor here. This branched off of this, which went down this route to make this common ancestor, but it could also have been going down this route to form this, and so forth. So this means we have another common ancestor here, which could have become this one, which becomes these 2, or it could have been, this and so forth. Down here, we can see it branches off into this, or it could have carried on into something else. So we have another common ancestor there. So here, each branch tells you a common ancestor, like so. So the question says, what do these classifications suggest about the evolutionary relationships between these 2 species of snake? So look at the differences between the 2 of them. This one here suggests they're more closely related, these 2. Whereas when you do it according to which Y was what their genetics, these 2 are more genetically related. So these appear similar, but these 2 are more genetically related. So then, classification X is saying that the L. maclotti and L. olivaceus are more closely related and share a common ancestor. Whereas here, this one shows this olivaceus and L. papuana to be more closely related and share a common ancestor instead. That's all they want to, that's all they care about. They do not care about anything else, right? Just look at what the question's saying. Suggest about the evolutionary relationships. Evolutionary relationships is all about how closely related they are. Do they share a common ancestor? How soon ago did they branch out? So it says here to fill out this table. So the way I remember it is desperate King Philip cried out for goodness sake. So for goodness sake, genus species would have been below it. So let me just go backwards. So out, cried, Philip, kingdom, desperate. So king, sorry, so it's going to be domain, kingdom, phylum, class, order. So there is a debate about the name of one of these species. Some people say it should be this, Papuana, or this, Papuana. So the way we write this is we always do genus and species. So the bit that they're disagreeing on is the genus. We usually use the binomial nomenclature for this, which means binomial, 2 named, or 2 thinged. So here, genus, species, that's how we write things. Like humans is Homo sapiens. Homo, genus, sapiens, species. So then it says, give 3 comparisons of genetic diversity that scientists used in order to generate classification Y. So remember, classification Y is the comparison of genetic characteristics. So what determines genetic characteristics? Well, number one is their DNA-based sequence. And then people are like, ah, dang, now what? Well, if you think about it, DNA is then transcribed into mRNA, which then eventually becomes amino acids and protein. So you don't have to do DNA on its own. Maybe you could, if they're gonna, make the same DNA or similar, they'll make same or similar mRNA. So then next you could look at their mRNA-based sequence as well. But then this eventually becomes the amino acids. So if they have the same amino acid sequence, yes, there are some differences in the base sequences, right? You would end up having similar, if not same, mRNA. So you could look at the amino acid sequence. But the problem with using that is obviously, remember, the DNA is degenerate, so multiple codons make the same amino acid. So you'll have multiple possibilities, and you can't exactly work out what the base sequence is, but at least you can have an estimate of how similar they are. mRNA is probably closer. The best bet is obviously comparing them directly. So this figure shows the development of an insect called a damselfly. The adult damselfly uses a tracheal system for gas exchange. Explain 3 ways in which it's adapted for efficient gas exchange. So I ain't gonna lie, why would I sit and memorize like 2 million facts when I can just think about the main things, right? So the main things that affect exchange, like I said a couple questions before, is concentration gradient, keeping it steep, short diffusion pathway, high temperature, which obviously we can't adjust to the living thing, so that one's out, and high surface area to volume ratio. So then all you need to do is apply it to that. So I'm going to choose short diffusion distance. Then we need to think, okay, well, tracheal system. So the tracheal insects, trachea branch out into tracheae, sorry. So then that means the tracheals are where, they're exchanging stuff into the cells. Short diffusion pathway means that they have, I'm going to say, well, if they have a short diffusion pathway, they must have thin walls. So that means there's a short diffusion pathway to cells. So you can just do it like that, and then it helps you sort of recall what you need, like what facts to bring out. Surface area to volume ratio, where we have large numbers of branched tracheals, which means you've got a large surface area for gas exchange. As well as that, concentration gradient, well, I mean, remember that abdominal movements moves air into the spiracles and out of them, so O2 comes in, CO2 comes out. So then, muscular movements like abdominal muscles move air in and out, and that maintains the concentration gradient. So a lot of people, what they did is they just listed things out and didn't say what they explained. So the features here, and then you say how it helps, because the short diffusion pathway, because of the branch tracheals, because of the concentration. They missed that latter part out. So the damselfly larva is a carnivore that actively hunts prey. It has gills to obtain oxygen from water. Some other species of insect have larvae in a similar size and shape and also live in water, but these do not actively hunt prey and do not have gills. Explain how the presence of gills adapts the damselfly to its way of life. So if we look at how it lives, it's chilling, it lays eggs, the eggs hatch and whatever, and you've got the larva here and they've got gills. So then it's told you here that these gills are in the larvae. And it's asking you, how are these larvae good at living in water? Right? So, in this example here, you can't just simply say something like, oh, well, they live on water, this and that, blah, blah, blah. Like, it's telling you that other larvae do not have gills and do not actively hunt prey, but they still also live in water. So why do these ones, need gills? Why can't they just do the same as the others? So that question's really asking you, gills versus no gills, both in water. So you really need to understand that. The only explanation that makes sense is that they are carnivores and they actively seek prey. So because they actively seek prey, that must mean that they have a higher respiratory rate and metabolic rate, so they're going to need more O2 in a given time. The whole point of gills is to, exchange gases and get more O2 in, CO2 out. So the only explanation for that is that they must be using more and, oxygen. That's it. A scientist measured the size of each gill lamella for 40 damselfly larvae, and the results are here. So we've got the mean width and mean length and the uncertainty. Calculate the mean surface area of one side of the gill lamellae. Assume that it's a rectangle and give your answer to appropriate number of significant figures, and then give the percentage uncertainty in your thing. So when width and length 1.61, 6.12, surface area of one side, this is the gill lamellae of 40. So what we're going to do is we're going to work our surface area first of all. So it's going to be 1.61 times 6.12. Right, so 1.61 times 6.12, which is 9.85 millimeters squared. Now, we don't need to worry about it being 40 because it's the mean width here, but just be wary that sometimes they may have the total, for example. Right, so that's your mean surface area, but they wanted to appropriate number of significant figures. They've given us 3, so we'll stick with 3. Cool. the uncertainty, so percentage uncertainty equals your raw uncertainty over your measurement. Now, because we're timesing two things together, when you're combining them, you need to add the percentage uncertainties. You never subtract them, you're always adding them, and you never times them either. So in this case, we need to work out our percentage uncertainties for each of them and add them together. So it's going to be 0.19 over 1.61 times 100 to get the percentage uncertainty for that one, plus 0.41 over 6.12 times 100. If we add those together, that'll give us our total percentage uncertainty, which is 100, 0.19 over 1.61 plus 100, 0.41 over 6.12. So our percentage uncertainty over this in this entire thing is 18.5%. So the mean surface area is 9.85 millimeters squared, and it's 18.5%. When you're timesing two different things together, you can't just add their uncertainties. You have to do the percentage uncertainties, because this is the uncertainty of this measurement. This is the uncertainty of this measurement. Because we're combining these two separate measurements. These are two different uncertainties for two different things. The only way to combine them is by adding percentage uncertainties. So then a student used an optical microscope to then look at the damselfly lava girl, and here's the figure. It says suggest two ways that they can improve the quality of this. So first of all, you can see down here this shading. We don't use shading. As well as that, what else can we do? Well, we have no magnification. So wait, sorry, we should really say no shading and then add magnification and scale bar. That's what screams to me first. Other things you could say is we shouldn't really cross label lines. They should be nice and clearly obvious. As well as that, they're sketching as well, so use single lines to draw stuff and make sure lines are continuous and connected as well. Apparently in the examiner report, some people were talking, enough people for them to cause a problem and write this in the mark scheme, was saying to use an electron microscope. This is a student, dude. I don't know what kind of schools you go to where you have electron microscopes, but nah, man, come on. And that's not what it's asking anyway. It's to draw the picture of the quality of the picture they drew. So then it says contrast how an optical microscope and a transmission electron microscope work, and then contrast the limitations of their uses. So just literally one by one, Transmission electroscopes, they use electrons, and optical uses light. What else can you know about terms? Well, the differences is that, they have a higher magnification and resolution because the electron wavelength is smaller than light. So then what does that mean? It means you can look at smaller organelles, right? But the optical can't do this. Limitations of terms. So we bashed on optical all the way there. Limitations of terms. Well, you can only look at dead specimens only. You have to do it in a vacuum, remember, because it needs to be done in a vacuum. Actually, let's add that. Otherwise, if you don't do it in a vacuum, the electrons will just smack against other atoms and electrons around, and it will just interfere completely and ruin stuff. There's no color here in TEMS, but optical can. As well as that, TEMS can only look at very, very thin, so very thin, like thinner specimens. Because remember, we're contrasting. We're comparing one versus the other. It's also, you need more preparation and expertise because it's more complex. What else can we talk about? I think I've rinsed it here, but other things you could talk about, for example, are like how they actually work as well. So like focusing lights as well. So optical uses glass lenses and TEMS focus with magnets, but like these are the things I'm saying. So figure 6 shows image of an optical microscope undergoing meiosis occurring in a flowering bud. W and Z are undergoing meiosis. Explain the appearance of W and Z. So if you look at W has got 4 cells, so therefore it must be at the end of the second meiosis division, right? And then therefore, if you look at W, we've got 4 haploid cells with N number of chromosomes. That's what it must be. That's why there's 4 of them, and that's why they're like this. So then if we look at Z, Z's got what? 2 cells? So therefore it has to be the first division. Here in Z, the 2 cells are haploid, but 2 times the genetic material. That's because the homologous pairs have separated. So a lot of people get this bit confused. Homologous means if I have my right arm here, that's your right arm. Our right arms are homologous, but one's from you, one's from me. in chromosomes, imagine if I look at chromosome on number 7, right? A normal diploid cell has this, maternal and paternal. Before mitosis and meiosis, these replicate to form double-armed sister chromatids. These are copies of each other. So we've got maternal and maternal, paternal and paternal. This is what happens at the start of meiosis. The very first division, these split. So then you've got, obviously after crossing over and all of that stuff, so you've got maternal, maternal, and then paternal, paternal here. This is diploid. We've got 2 times the amount of diploid stuff. You can't say 4n because that implies you'll have 4 different parents. So like maternal, paternal, something else, and something else. That's why you say 2 times 2n because there's double it, right? Here, the homologous pairs are the same chromosomes from respective parents. So the maternals and the paternals have split. So the homologous pairs have split. So now you've got double the amount of n haploid because there's only maternal, only paternal. Then what happens? These sister chromatids then split. Like this. So we have maternal, we have maternal, we have paternal, paternal. Remember that there's crossing over and all of that stuff happening to make this more diverse? And then you've got n in each of these. They've not got doubles again. That's what happens in meiosis. That's the homologous pair bit separating, which makes you haploid. Mitosis is different because in mitosis you are diploid to start with, so maternal, paternal, not together with a sister chromatid. Then you replicate you're replicating your DNA. So then you'll have these 2 sister chromatids, maternal, maternal, and then paternal, paternal here, like this. And then what happens is the daughter cells eventually end up with a maternal and paternal of each. There's no homologous pairs separating because both of them still have homologous pairs, right? These sister chromatids are not maternal and paternal. They are doubles of the original there. It's really important you drill that into your head because I see it all the time where people talk about all kinds of yap and it's... So then, an environmental scientist investigated the possible relationship between air pollution and the size of seeds produced by one species of trees. He's provided with a very large number of seeds from a population of trees in the center of a city and then also a large number of seeds collected from a population of trees in the countryside. Describe how you should collect and process data from these seeds to investigate whether there is a difference in seed size between these 2 populations. Independent, dependent. What are we going to be doing then? So we're looking at... So what are we changing? We are changing the environment the trees are in. And the dependent is what we are measuring, which is the seed size. So we're going to collect lots and lots of different ones, and we're going to work them out. So what you need to do first is you need to collect random samples of seeds from each population in each environment. Then what you want to do is you want to measure them. So we're going to measure their mass of each sample and work out the means. Then we want to see if there's any difference in the seed size, right? So we're going to be comparing the mean mass. So what you're going to do is you're going to calculate a p-value using the t-test to see if, and the p-value is testing whether there's a difference in the mass of the mean mass between the two, to see if there is a significant difference. You can't just say in the results or whatever, you need to say what it is in the mean mass of seeds between the populations. And there you go, that's it. So the t-test, any p-value, you need to decide what that p-value is testing. This p-value is testing the difference in the means masses between the two. So group 1 has got a mean here, group 2 has got a mean here. This might be, I don't know, 10 and this might be 5. It's looking at whether the difference between these two is actually there and if it's significant, not just the results. So there you go. Thank you very much. Please like, share, and subscribe. You know the drill. And yeah, thank you.